as a Government of Canada research scientist, it's implied that I have no conflict of interest. And so, so, so I want to talk a little bit about our work on lentils, and with the assumption that there's good evidence that whole lentils are good food and they lower, like other pulses, lower blood glucose response. And I think the solution to the pasta problem in Italy would be to add Canadian lentils to the diet, and I would sort that out. So the question though is, are the pulse products or flours derived from lentils able to confer the same health benefit? And what is the effect of processing on lentil and blood glucose response? I want to talk a little bit about the studies we've done on relating lentil starch fractions to blood glucose response, and then look at the effect of sample preparation on starch fraction measurements, and then perhaps some of the next steps and areas to consider as we move along. So when you look at the literature and focusing only on pulse flours, you see here a study published in 2012 from the group in Guelph. Spray-dried pulse consumption does not affect cardiovascular disease risk or glycemic control in healthy males. And then you've got this paper published by Harvey's group last year. No differences in blood glucose response were observed between the pulse treatments due to processing between whole canned pulses, pureed canned pulses, and pulse powders. So no effect, and in here, in this study, the pulse powders were able to maintain the blood glucose lowering effect. So I think we've identified some of the gaps, and one of them is we need to understand better the mechanism by which pulse improved glycemic control. We know that disruption of the starch cellular structure influences digestibility and therefore the postprandial blood glucose. And there's this need for structure function studies, and this is where we begin. So we wanted to do a human trial, a 12-week trial in which we were trying to assess the efficacy of whole lentil products in lowering the 2-hour postprandial blood glucose. And we weren't sure which product to start off with. So we had cooked whole lentils. We had cooked lentil puree, freshly cooked lentil puree. We had a cooked spray-dried lentil powder or flour. We had a roasted lentil flour, and we compared all of these to an instant potato control. So in a GI study, together with Tom and GI Labs, we showed that this one lentil variety processed into 4 different forms. So one lentil variety processed to 4 different forms, fed to 10 human volunteers. You see that there's a range of glycemic response. So whole lentil, freshly cooked lentil puree, frozen lentil puree, roasted lentil powder, but here you have the spray-dried lentil powder. So as you can see, as we process the lentil, we change the postprandial blood glucose response. Here's white bread, and this is the instant potato. So we wanted to explore this a little bit further, and we showed that there's a relationship, and this is not novel, but the glycemic index of the different products is related to the rapidly digestible starch positively and negatively to the slowly digestible starch. No surprise there. So we then designed a study to look at the relationship between lentil starch fractions and postprandial blood glucose response in different lentil varieties, Canadian lentil varieties. And the aim here was to identify component in lentils responsible for lowering blood glucose response. So we wanted to define the chemical composition and starch fraction of the cooked lentil varieties, to characterize their physiochemical properties, and then to conduct an acute human feed-in trial to assess postprandial blood glucose response against the different slowly digestible starch fractions. So we started out assessing the starch fractions in 20 varieties of Canadian lentils. And as you can see here, this is a total starch. The red shows the slowly digestible starch, and the green, the rapidly digestible starch. So we were able to then take a variety with the highest slowly digestible starch, and then one with the lowest slowly digestible starch, and then 6 in between here, and do a human GI study together with Tom. And this is what we found. No real differences in the postprandial blood glucose, certainly different from white bread. So all varieties had lower postprandial blood glucose than white bread. And the postprandial blood glucose response elicited by the different varieties were not significantly different from each other. But when we plotted the in vivo glycemic index against the slowly digestible starch, we saw this, again, we saw this inverse relationship. We also saw a positive relationship, again, between the in vivo glycemic index and the rapidly digestible starch. Interestingly, there was no correlation between glycemic index and the resistant starch. So we wanted to explore this a little bit in more detail, and of course we can do this faster if we're doing it in the lab. And we got this instrument from Cyro from Australia, marketed by Next Instrument, and that's not an advertisement. But we were able to adapt the English method onto this method, and essentially you can run 20 samples with this is auto sampler here and an onboard glucose analyzer. And so we validated the method on this system, and so we've been looking at the relationship between in vitro starch digestibility and processing. And here you see some of the preliminary results. Here, these two lines here show first, so we cooked the lentil, freeze-dried and ground it into a powder, or oven-dried, grind it into a powder, and digested it. And you see this pattern here is not the sort of in vivo response we see. It's quite a rapid digestion. Whereas here, where you have the freshly cooked lentil made into a puree and digested, you see this sort of response we see in vivo. And here, this is lentil that was roasted in a convection oven and ground it into a flour. So you see processing, we're finding here that processing and heat exposure, milling, all affect the digestibility, at least the in vitro digestibility of the same lentil sample. And microscopically, we can explain some of this by the starch granules. So here you see the starch granules are intact within the cell wall, and most of it gelatinized. This is non-gelatinized. Here, this is oven-dried lentil puree that's been milled into a flour, similar to the freeze-dried, cooked freeze-dried and grounded lentil, and lack mostly gelatinized starch. When you look at the roasted lentil, though, the starch granules are outside of the cell wall. But but most of the starch is not gelatinized, as evidenced by the Maltese structure there. So when you reconcile this with the digestion profile, I think we start getting at some insights into the effect of processing on the in vitro digestibility of lentils. So to summarize, processing affects in vivo digestion profile. Whole pulse and puree have the lowest postprandial blood glucose response, and the roasted pulse flour had a lower postprandial blood glucose response. We found that after boiling, pulse starch granules become swollen and partly gelatinized, but retain their integrity during in vitro digestion. We found that the pulse flours lose structural integrity with grinding unless the pulse is roasted and then ground. And I think it's safe to conclude that postprandial blood glucose response may be explained by starch gelatinization and cell wall disruption, and this is related to slowly digestible starch and rapidly digestible starch content. And moving forward, I think we need to compare like with like, and we need to ensure that the postprandial, we need to ensure that the postprandial blood glucose reducing ability of whole pulse is retained during processing into flours. We can do this perhaps by growing new varieties, by ensuring that the methods that we use to produce pulse flours allow the pulse flours to retain the ability to reduce blood glucose response. In terms of some of the in vitro work, we want to try to develop a method that allows us to mimic mastication. And really, what I think one of the gaps in this work is being able to compare foods that are as eaten in the in vitro system. So I think we need to be comparing like with like and apples with apples. So I think we will continue to measure the in vitro digestibility profile and starch fractions of the samples as eaten, and that's a challenge, because when you cook, freeze dry, and grind the powders, the digestibility profile changes. And of course, we need to continue to look at the structure-function relationships by doing human trials to assess the effect of processing on the in vivo postprandial blood glucose response. Thank you.
Really a comment. I want to say studies like this are needed for every grain and every type of grain and legume that we're consuming, because the progress that we have in food processing has really pushed us further and further up the GI scale. But your sort of research is telling us there are tricks that we can use to, and the same with the pasta presentation earlier. There are tricks we can use to lower the GI and still have the same palatability and other characteristics that the consumer wants.
Absolutely, and I mean, if you look at these kind of studies, I think, and thank you for your comment, I think these sort of studies allow us to think of new ways to incorporate more pulses into more products and make them more market available in the market. The study published by Harvey's group last year, if you look at the methodology, it becomes clear that the reason why the pulse flours maintain the ability to reduce postprandial blood glucose was because they used something called micronization. It's an application of a very high heat for a very short period of time, and then milling the pulse into a flour.